Hello friends, today we are going to discuss about glycolysis. Glycolysis starts from 6 carbon sugar, that is glucose. Glucose splits, that's why we known is as a glycolysis. Glyco means glucose and lysis means splitting. Particularly first the glucose is split, glucose is added phosphate group at 6th position from the breakdown of ATP. ATP's third phosphate bond is break and this phosphate is added to 6th position of glucose, and hence in first step we got glucose 6 phosphate and phosphate is added, hence the reaction is known as phosphorylation. This reaction is catalyzed by an enzyme hexokinase. Hexokinase works in presence of Mg++ ions. Then the second step of this reaction is the isomerization. Isomerization of the glucose 6 phosphate into fructose 6 phosphate. This reaction is catalyzed by phosphoglucoisomerase. There are 1 molecules in each step of the reactions, but at the end of glycolysis we got 2 molecules of pyruvic acid or pyruvate. The glycolysis is same or more or less same in all species, but here we are going to discuss about the aerobic respiration particularly. That's why I say the pyruvic acid. Then in third step of this splitting reaction is one another phosphate group is added at the first carbon position of fructose, hence we got fructose 1,6 diphosphate, which is also a 6 carbon molecule. ATP is broken down, we'll get ADP, and the third phosphate is added at the first position of fructose. This reaction is also known as phosphorylation, and the reaction is catalyzed by phosphofructokinase in presence of magnesium plus plus ions. Then in fourth step, the fructose 1,6 diphosphate is splits into two 3 carbon molecules, that is dihydroxyacetone phosphate DHAP and PGAL. This reaction is known as cleavage. At this position, our first phase, that is the preparatory phase of the glycolysis, is ends up. We'll get 2 molecules of each 3 carbon containing. This reaction is catalyzed by aldolase enzyme. These are isomers of each other, hence DHAP readily converts into PGAL. Hence we got 2 molecules of PGAL, that is phosphoglyceraldehyde. Then the phosphoglyceraldehyde comes and will get 2 molecules of each 3 carbon phosphoglyceraldehyde. Then all the reactions takes place on the phosphoglyceraldehyde. After the isomerization reaction of DHAP and PGAL is catalyzed by triose phosphate isomerase. Then after the isomerization of PGAL, we got 1,3-diphosphoglyceraldehyde. This 1,3-diphosphoglyceraldehyde also has 2 molecules. In glycolysis there are 2 phases. Here the first phase, that is the cleavage phase or preparatory phase, is completes up at the up to the fifth reaction. After the fifth reaction we are enter into the synthesis phase, where exactly the ATP molecules are synthesized by the stepwise lysis or stepwise manners of reactions. Particularly the sixth reaction is oxidation, where the NAD is reduced into 2 NADH2 molecules. Here from we got a 2 molecules, that's why we got a 1,3-diphosphoglyceraldehyde. Here the phosphoric acid, that is H3PO4 group, is added. This reaction is catalyzed by phosphoglyceraldehyde dehydrogenase. After the 1,3-diphosphoglyceraldehyde, one group is removed and one phosphate is removed particularly, and we get a ATP molecule from the ADP. In this reaction, first after the oxidation, first ATP generation is carried out. This first ATP generation takes place here, and we got 3 phosphoglyceraldehyde molecule. ATP generation first is catalyzed by phosphoglycerokinase in magnesium plus plus ions. Two ADP molecules are added, and these from two ADP molecules, two phosphates are removed from the first position of diphosphoglyceraldehyde. We get two ATP molecules, and we get here first time two ATP molecules, and not these are required, but get. That's why after the fifth reaction we say it is a synthesis phase. Here energy is synthesized. Then 3-phosphoglyceraldehyde readily changed, particularly here the shifting is takes place and phosphate group is shifted towards the second position. We get a 2-phosphoglyceraldehyde. That's why the reaction is popularly known as shifting, and this shifting is carried out by the enzyme phosphoglyceromutase. Here also there are two molecules of 2-phosphoglyceraldehyde. After that 2-phosphoglyceraldehyde, the ATP dehydration takes place. In dehydration, H2O molecule is removed from 2-phosphoglyceraldehyde, and we get phosphoenol pyruvic acid or phosphoenol pyruvate, two molecules of phosphoenol pyruvate. Simply we say this phosphoenol pyruvic acid as PEPA, that is PEPA. Here H2O molecule is removed by an enzyme enolase in Mg++ ions. After the formation of phosphoenol pyruvate or phosphoenol pyruvic acid, this phosphoenol pyruvic acid readily converts and ATP generation takes place. Here the two phosphates are removed, or particularly from two molecules each of one phosphate is removed, and 2 ADP gives us 2 ATP molecules, and the phosphoenol pyruvates gets convert into pyruvic acid or the molecule of pyruvate. Hence we say that after the fifth reaction, that is the isomerization, there is synthesis of ATP is carried out. There are total number of 4 ATPs are synthesized, 2 at the 7th reaction and 2 at the 10th reaction. Rather than that, in 6th position, NAD is also reduced and 2 NADH2 molecules are prepared. Hence, from each NADH2 molecule, we get a 3 ATP. That's why total number there are 6 ATP are synthesized, 6 ATP there, and 2 and 2 4 are total. That's why the total number of ATP are synthesized in glycolysis are 10. And on the other hand, in the preparatory phase particularly, or the cleavage phase, 2 ATP molecules are used up. Hence the net gain of ATP after the completion of glycolysis is remains 8 ATP. We say here we see here in the slide particularly the how much ATP loosed at the preparatory phase and how much we get an how much we get total number of ATP 6 and 4 10 ATP we get. Loose of ATP is 2 ATP. That's why the net output or the final output of glycolysis